In Duchenne muscular dystrophy, muscles break down faster than the body can repair them. Over time, muscle damage outpaces the body's ability to repair itself, leading to progressive muscle loss. In healthy muscle, stem cells divide in a balanced way, creating both new muscle cells to repair damage while retaining stem cells to keep the process going in the future. Satellos discovered that in Duchenne, this balance is lost. And why? Simply put, we found that stem cells don't divide properly, making mainly new stem cells instead of new muscle cells. This means the body can't create enough new muscle cells to keep up with the ongoing damage. At Satellos, we also discovered how this process is failing in Duchenne. An important signal that muscle stem cells need is missing. Normally, this signal is provided by a protein called dystrophin, found in those muscle stem cells. We found a way to replace that missing signal in Duchenne by blocking a completely different protein from an alternative signaling pathway in the body. This approach helps bring stem cell divisions back into balance, allowing the body to resume repairing muscle. We're developing SAT-3247, an investigational oral drug treatment that we've designed as an inhibitor of this protein called AAK1. Our research has shown that inhibition of AAK1 with SAT-3247 can reset balanced muscle stem cell division in response to damage. This has the potential to restore the body's natural muscle repair process and rebuild muscle. Our goal is to restore the body's natural ability to regenerate muscle from within. In Duchenne, where muscle repair is disrupted, we aim to restart the cycle of muscle growth and renewal. Learn more at Satellos.com.